Hi, welcome all of you. My name is Dinesh Chauhan and today we are discuss about the types of DNA. So let's start. So the content what we discuss here. So first what is DNA, then we discuss the what is the type of DNA, then we discuss the type or forms of DNA, that is B DNA, A DNA, Z DNA, C DNA, D DNA, E DNA, then the summary and the reference. Next, what is type of DNA first. So DNA is a genetic, we know that a DNA is a genetic material. It holds all the information of making protein. Or we can say that DNA is genetic material that transfer from one generation to next generation. But what does mean? So to understand this, we want to see this experiment. So DNA is a genetic material that we see and it transfer from one generation to next generation. Means actually what? So that we understand here. Suppose we say for that we want to see this experiment. So we have two bacteria, one is R10 bacteria and other is S10 bacteria. So R10 bacteria have no cell wall around their membrane, and the S10 bacteria have the cell wall around their membrane. so and here we see the test tube that contain the culture. The test tube contain the culture of R10 bacteria, nothing but R10 bacteria. In this test tube we add the DNA, only the DNA of S10 bacteria. Actually this DNA or the gene have the information about the production of capsule. So this gene is added into this culture of R10. So when we add this DNA into the culture of R10, this DNA uptake, this R10 bacteria uptake this DNA into them and convert this R10 bacteria into the S10 bacteria. How? Yeah, we already told that this DNA have the information about the production of DNA. So this DNA when accepted by the uptake by the R10 bacteria, so they also now become capable to produce the capsule, and they convert, and that's why they convert into the S strain. Now our question is that DNA genetic material transfer from one generation to next generation. We also not get an answer of this question. So what the answer is, this is a S10 bacteria. This contain this this contain the DNA of S10 bacteria. So this is suppose this is first strain of bacteria. This first strain, this is the first generation, and when this is the parent cell, and when this convert, and when this parent cell is undergo the division, they form the two cell, and this two cell, nothing the daughter cell, this also contain the same DNA or same gene, and they also have the capsule. Means only arsine bacteria is unable to produce the capsule, or they not have a capsule, but due to this they uptake this DNA, they produce a capsule. Now these S10 bacteria, means R is converted to S, this S10 bacteria also have this strain, this new S10 bacteria also have DNA. If the daughter cell, if this gene is not transferred to daughter cell, then daughter cell, if not transfer, then daughter cell not produce the capsule, but it transfer, and that's why the capsule is produced or is also around the daughter cell. So that is a reason that DNA is genetic material, and what the meaning of that is this. Next is next that DNA, actually structure of DNA. Next the structure. So DNA is actually what actually is consist of the is a polymer of nucleotide. So two chain of nucleotides are coiled around each other and form the duplex, form the double helical structure. Okay, and this nucleotide is composed of a nitrogen base, sugar and phosphate. So nitrogen base either the guanine, cytosine, adenine or thymine. Okay, this is all about the DNA. Then we discuss about the types of DNA. What is the type of DNA? Yes, what is type of DNA? So type of DNA means what? Actually, as we we learn in a 12th or 11th that structure of DNA as like these bDNA, like this DNA. But there are also the different types of DNA. Now this question is quite confusing, that there is a type of DNA. Yes, there is a type of DNA. How that? So depending, actually structure of DNA is not fixed. DNA structure is changed according to the condition around the DNA. So what is the definition of type of DNA? So when what is so depending on a condition, the structure of DNA is change change. That is called the type of DNA. So listen carefully. Depending on a condition, the type of DNA, depending on the condition, the structure of DNA is change around the DNA. So depending on a condition around the DNA, the structure of DNA is change. For example, suppose if the condition around the DNA is a high water or high humidity, then DNA exists in a B form. If the condition around the DNA is low humidity, then DNA exists in a A form. And the most common form of DNA is the bDNA. Okay, then we see the different forms of DNA like A, B, A, C, D, E and Z have relieved after X-ray diffraction analysis of DNA crystal at atomic resolution. So all these structure are relieved, come after the X-ray diffraction analysis. Next we want to see the types or forms of DNA. There are actually 6 forms of DNA or types of DNA. Most so bDNA, zDNA, aDNA, cDNA, dDNA and eDNA. When someone asks you what is the type of DNA, then you say A, B, C, D, E, Z. But during the learning, we want to learn first B, then Z, then A, then C, then D and then eDNA. That is most easy for the learning. Okay, so next we want to see the bDNA. First is a bDNA. So in all these, what we see for each of these DNA, we want to see for each DNA their condition, in which condition they exist, or which condition they exist, and their characteristics, means how their base pair per turn, then their and then their then then their most of the then their structure is how arranged, their nucleotide bases are how arranged. All these things we have to see here. The first is a bDNA. So what condition require bDNA? So bDNA is present in every cell at a very high relative humidity and low concentration of ions. We already see that during the high temperature, during the high humidity, the DNA exists in a B form. And this DNA, now the characteristics. So this DNA is in a right-handed double helix, then 10 base pair per turn of the helix, then is proposed by the Watson and Crick. And this structure is proposed by Watson and Crick, and that we are learned in 11th or in 12th. Actually, when the Watson and Crick performed the experiment to prove the structure of DNA, at that time there is also the high humidity condition. In a high humidity condition, they perform that experiment. That's why DNA structure is they found in a B form. Actually, in our body or in a mammals and in a plant, all the DNA in a cells are bDNA, because in our body we know that our body or cell contains 70% of water. So that's why in our body there is a B form of DNA exist. And then base pair are perpendicular to the longitudinal axis of the helix. Then B forms of DNA is metabolically stable and undergo the change to A, C, D forms depending upon the sequence of nucleotide and concentration of access salt. So this DNA may changes, means what? If the in case of there is a, instead of low concentration of iron, if there is a high concentration of iron, then also the DNA structure is changed. Okay, So depending on condition, structure of DNA is changed around the DNA. So depending on the condition around the DNA. Next, the aDNA, and as usual we can see the aDNA. So what the condition require the DNA to exist in A form. So require the 75% means relative humidity, means low humidity and presence of sodium ion, potassium ion and cesium ion they require. This then the characteristic is a right-handed double helix, then 11 base pair per turn per turn of helix. Now the helix means, if you know, the helix means what? The portion from here up to this is the helix. This is a helix. Okay, portion from this to this is the helix. This is helix from here to here. This is the helix. This is all is a helix. Then similarly we can see about a zDNA, similarly for the cDNA, similarly for dDNA, similarly for eDNA. This is, you might be bored that I say that, so only we discuss the condition and the characteristics. So we come next is the zDNA. So zDNA require very high concentration of, the zDNA require a very high salt concentration, and zDNA is only in a left-handed direction, in a left-handed coiling. Then base pair per turn of helix is 12. Then proposed by this zDNA is proposed by the Alexander Reich, two scientists. Then special feature is of zDNA is that B and Z forms of DNA are interconvertible. Then base pair is arranged peripherally to the helix. Next, the cDNA. The cDNA, what condition require? 66% humidity and presence of lithium ion. If this condition is present around the DNA, then DNA exists in a C form. And then coiling right-handed base pair is 9.3 per. Then the proposed no. Then the occur in vitro, arranged toward the middle of helix. Then the dDNA is a very rare. dDNA is very rare, very rare. Then is coiling is right-handed. Then base pair per turn of helix is 8. Then the special feature require, here the special feature, DNA is locked. dDNA is lack the guanine, nitrogen base, lack guanine. Then arranged, then base pair is arranged toward the middle of the helix. Then the eDNA require eDNA when the condition around the DNA is induced. So there is a not condition here, that the induced by the cytosine and methylation. So eDNA structure induced by cytosine methylation or bromination. If the cytosine is a nitrogen base, so when the cytosine nitrogen base, when methylation of cytosine or bromination of cytosine is takes place, then this DNA is exist, or this DNA is induced. Here the here the base pair require 7 point, here the base pair is a 7.5 per turn of helix. Means what? In every turn, means this is a helix, means base pair is also not fixed. Then it occur in vitro. eDNA is occur in vitro, means what? This DNA you not see in any cell. This DNA is only in a DNA, only in a lab we can see, or during the experiment. Base pair is perpendicular to the helical axis. So this is all about DNA. This is all about the type of DNA. Actually, I collect information from the books, from the internet. I get a very less information. Even I contact with the teachers that I know, all I contact, and then all the information, whatever I get, I write it down here, and I write it down, understand that, and then present this video. Thank you. If you like, subscribe my channel and support our channel.